Welcome in BSc Studies Classes channel. So this is my second video. In the previous video we are discussing about the taxonomy and angiosperm, and in the today's lecture we are going to discuss about the most important topic mentioned in the botany. This is the plant, tissue and culture. So plant, tissue and culture, these three words is the most important in the botany, because this plant tissue culture is the magical science we are utilized for plant tissue culture to reproduce or produce the whole plant body from the single cell. Okay, so very big thing, this whole plant body we are produced from the single tissue or single cell. So let us see what is the plant, tissue and culture. So plant, tissue and culture, these three words are utilized in the plant tissue culture, and plant tissue culture is also known as micropropagation. Plant tissue culture is also known as micropropagation. So micropropagation means there is the two words. First is the micro and second one is the propagation. So micro means very small or very smallest particle, or this is known as tissue. Okay, tissue and or the cell is very small, means the tissue and cell you are not observed by your naked eyes. So this tissue or cell is observed only microscope. And second one word is the propagation, means to reproduce the new individual from the micro tissue or micro particle, means cell. So this is also known as micropropagation. But what is the meaning of plant, tissue and culture? So first word culture, second word is tissue and first is the plant. Culture, tissue and third one is plant. Okay, so culture means to growing or to grow or grown the new plant body. Means in the farm, by the farmer, they are harvesting the different types of the plants or crops in the farm. This harvesting method is known as growing method, and this growing method is also known as the culture. This is the one of the type of the culture. So by using the culture, we are utilize the culture to reproduce the new individual plant body in the particular located area, is known as culture. But in the micropropagation, there is an artificial modification system in the botany, and we are utilize this artificial method for reproduction. This is known as plant tissue culture or micropropagation. So culture means to grow to the plant body. There is the second one is the tissue. Tissue is the very smallest part, or this is also known as group of cell. So many cells, they are come together and formation of one layer. This layer is known as tissue. So many cells, they are compactly arranged and they are formation of the tissue in the plant body. For example, you have to go on the field and in the particular located area and observe any plant body. You have to observe the plant body, for example, part of the plant for stem, leaf, flowers, etc. So this part you have to observe by externally by morphological characteristic feature. Means by the macroscopic character, you have to observe the macroscopic characteristics only. means morphological characteristics only, but from the macroscopic characters you are not observe which tissue they are present inside the part of this particular uh, part of the leaf plant. For example, leaves, you are not conclude that which type of the tissue system are present inside the particular part of the plant. So for the observation of the cell or tissue, you have to taking the transverse section of the particular plant body or particular part of the plant body, and this section is observed under microscope. So we conclude that which type of the tissue system are present or arranged inside the particular part in the plant body. Okay, so these cells, there are many cells, they are come together and formation of group, and group of cell is also known as tissue. By using tissue, we are culturing the plant, means we are reproducing the new plant body by using the tissue. These are the most important artificial reproduction method. And by using for the artificial reproduction method, we are utilize the living organism like plant. We are utilize the plant for the tissue culture technique. We utilize the plant for the artificial reproduction method by using micropropagation. Okay, so this is known as plant tissue and culture. So plant tissue culture is the most important modern technique in the botany. They are artificial reproduction. We are utilize the plant tissue culture where we are producing the new plant body from the mother plant one or from the tissue of the part of the mother plant. Okay, so we are discussing about the tissue culture in detail. So in the first step in the plant tissue culture method is selection. So selection of plants part is most important. First step is selection method, because which part you have to select for the tissue culture method, these are the most important, because first and most important criteria for selection is this part you have to observe in the field and you have to select for the tissue culture. This part is should be healthy or disease free. Healthy or disease free means disease resistance capacity they are present inside the part. Okay, so this part is most important for our baby plant. So this part is should be healthy or disease free. This is the first criteria in the selection method. So for example, this is first plant we are selecting for the reproducing the new plant body by using tissue culture method. This selecting plant is known as mother plant. So this mother plant you have to select, and this mother plant should be healthy or disease free. This is the first criteria and most important criteria. So by using the part of this plant, we are producing the new individual plant body or new baby plant from the using the mother plant. So this first plant is known as mother plant. So these plant are present. First is the root system. First part is root. Second one is the leaves. Third one is the flower. So this all part present inside the mother plant. So first and most important task is selection of part present in that mother plant. Which part you have to select from the mother plant is most important, because first is the root system or root. So root are not possible to select for tissue culture. Why? What is the reason? Because root is present in the underground portion and you cannot select root for the tissue culture method because this is the present in the underground portion. You are not observe root system in the field. So second one is the stem. Stem are utilized for the tissue culture, but soft target is the leaves or leaf system or in the mother plant. So you are selecting the leaves from the mother plant for the tissue culture method. Next one is the flower. You are selecting the flower for the tissue culture method, but the problem in the flower selection is the flower is the seasonal. They are not present in the all season. So this is the big problem to selection of flower for the tissue culture method. So soft target is the leaves. So you are selecting the leaves for tissue culture method. So for the tissue culture, you are selecting the one part of the leaf, not entire leaf. You are selecting one part of the leaves or leaf. This part is cut and separate from the mother plant. Okay. So listen carefully, this point is most important. This part of the leaf you have to separate from the mother plant, and this part is separated from the mother plant. That's why this part is known as explant. Why? What is the meaning of X? You are very well know this word utilized in the day-to-day life, ex-girlfriend or ex-wife. So means this part is present before inside the mother plant, but now we are separating this part from the mother plant. Then this is the first step, selection of explant. Then with the help of the machine you have to cut explant into small pieces. So this explant is cut into small pieces. So this explant is cut into small pieces. In the petri dish, we are taking the explant. From the petri dish, we are selecting one explant. And this explant is transferred into the test tube. We are taking the test tube for the tissue culture technology. And this test tube is made up from the glass. Okay. So this explant is transferred into the test tube, and inside the test tube we are taking the one of the most important medium is MS medium. MS medium is the liquid medium. For the growing this explant, they are required more amount of the nutrient or more amount of the required nutrition. For the growth and development of the explant, they are required more amount of the nutrient. Means in the natural harvesting methods or natural growing plant in the field, this plant they are absorb this all type of the nutrient or nourishment from the soil. But here we are prepare the whole plant body in the test tube without soil. This is the artificial method, and that's why we are providing the nourishment or nutrition or nutrient from the artificial media. This artificial media is known as MS medium, and this MS medium was prepared by the scientists Murarshige and Shuk. So by using MS media, we are transfer the explant into the medium. This transfer process is known as inoculation. Transfer process, transfer of the explant into the medium is known as inoculation. Don't get confused between inoculation and incubation method. So first step is the inoculation, means transfer of the explant into the medium is known as inoculation in the MS media. Then after transferring of MS medium or explant into the medium, we are required some periods or we are required some days for waiting period. So this waiting period is 20 to 25 days we are waiting for plant is growing inside the test tube. So this waiting period is, this step is known as incubation. What is the difference between inoculation and incubation? So inoculation means explant is transferred into the medium or explant is transferred into the test tube. In the MS medium is known as inoculation, and we are provided the waiting period for the growing the plant into the test tube. This waiting period we are providing 20 to 25 days. For example, this waiting period is known as incubation technique. After incubation, inside the test tube, small root systems is growing, and also they are growing the shoot system. Means baby plant are growing or growth and development of the baby plant in the after incubation technique. This plant is baby plant or they are in the growing stage. So now this plant is transferred in the, they are not transferred in the direct field because this plant is baby plant, so they are transferred in the greenhouse technology. And this transferring method is known as hardening. So this technique is known as tissue culture. Means by using one part of the leaf, we are growing the one plant body. We are growing the baby plant from the single tissue. But this explant is not one. By using one part of the leaf, we are prepare the 100 explant, for example. And is by using 100 explant in the 100 test tube, we are prepare the 100 baby plants from the one mother plant. This is the magical artificial propagation science. For utilize this artificial propagation, we are increase the number of the rare plants. Those plants they are rare, those plants they are going towards the extinct stage. So we are utilize the plant tissue culture, we are utilize the micro propagation, we are increase the number of the extinct endangered species or rare plants. For example, Ceropagia. Okay, so this we are increase the number of this plant, means from the single part of the mother plant, from the one plant, by using one plant we are prepare the 100 new plants. Okay, so this technique is known as plant tissue and culture. But the question is, what is the science or what is the magical science present in that plant? Why new plant body are developed from the single tissue? Why new plant body are developed from the single cell? This is the question. Which capacity present in that cell? Which capacity present in that tissue? By using this capacity tissue, by using this tissue, they are prepare the whole plant body. So this is technique is known as totipotency. Totipotency is the technique, is ability to develop the new plant body from the single cell. The cell, their ability to develop whole plant body, develop new individual, develop new living organism. This capacity or this ability is known as totipotency. So totipotency is observed in the all plants, but they are not observed in the all animals. Okay, for example, you are very well know starfish. Starfish, they are carrying the 5 arms and one arm are separate, for example, from the starfish. So they are developing or growing the new arm. Next example is lizard. Okay, so they are showing totipotency, but not all animals they are showing totipotency, but all plants they are showing the concept of totipotency. Means inside the plant, in the anatomy of the plant, the tissue or group of cell, their formation or their ability to form new individual plant body from the single cell. So this tissue culture technique is happening by their ability of the cell. Means the totipotency is the base of plant tissue culture. So this technique is known as plant tissue and culture. Means by using the artificial propagation, by using the micro propagation, we are providing the MS media by artificially manner, and we are growing or developing the new plant body from the mother plant. Okay, thank you.